स्टूडेंट्स, आज प्लांट किंगडम की एनसीईआरटी कवर करते हैं। अगर आपको प्लांट किंगडम का नोट्स चाहिए होगा और शॉर्ट नोट चाहिए होगा तो टेलीग्राम चैनल को ज्वाइन कर ले, जो कि लिंक दिया हुआ है मेरे प्रोफाइल में। चैनल के ऊपर जाएंगे तो, और अगर 20 ईयर के PYQs भी चाहिए होंगे इस चैप्टर के तो वो भी अवेलेबल है टेलीग्राम चैनल पे, वहाँ से जाके आप ले सकते हैं। ओके, लेट्स स्टार्ट द चैप्टर।
In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whittaker 1969, wherein he suggests the five kingdom classification: Monera, Protista, Fungi, Animalia, Plantae. In this chapter, we will deal in detail with further classification within the kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and the member of the Monera, Protista, having cell wall, have now been excluded from the Plantae through early classification, placed them in the same kingdom. So the cyanobacteria that are also referred to a blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophyte, pteridophyte, gymnosperm, angiosperm under Plantae. Let us also look at classification within the angiosperm to understand some of the concerns that influence the classification system. The earliest system of classification used only gross superficial morphological characters such as habitat, color, number, shape of the leaf, etc. They were based mainly on the vegetative character or on the androecium structure system given by Linnaeus. Such systems are such systems were artificially. They separate the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to the vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against these, natural classification systems developed which were based on natural affinity among the organism and considered not only the external feature but also internal feature like ultrastructure, anatomy, embryology, phytochemistry. Such a classification for flowering plant was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification systems based on evolutionary relationship between the various organisms are acceptable. This assume that organisms belong to the same taxa have a common ancestor. We now use information for many other sources to help resolve difficulty in the classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computer, is based on all observable characteristics. Now, number and code are assigned to all the characteristics and the data are then processed. In this way, each characteristic is given equal importance and at the same time, hundreds of the characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy that use the chemical constitute of the plant to resolve confusion, are also used by the taxonomists these days. Algae. Algae are chlorophyll-bearing, simple, thalloid, autotrophic, and largely aquatic, both freshwater and marine organisms. They occur in a variety of other in a variety of habitat, moist, stone, soil, and wood. Some of them also occur in association with the fungi, lichen, and animal. Example on sloth bear. The form and the size of algae is highly variable, ranging from colonial form like volvox and the filamentous form like Urothrix and Spirogyra, figure 3.1. A few of marine forms such as scalp form massive plant bodies. The algae reproduce by vegetative, asexual and sexual method. The vegetative reproduction is by fragmentation. Each fragment develops into a thallus. As asexual reproduction is by the production of different type of spores, the most common being the zoospore. They are flagellated, motile, and on germination give rise to new plant. Sexual reproduction takes place through fusion of two gametes. The gamete can be flagellated and similar in size as in eulothrix, or non-flagellated, non-motile, but similar in size as Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size, as in species of Eudorina, is termed as anisogamous. Fusion between one large, non-motile static female gamete and a smaller, motile male gamete is termed as oogamous. Example: Volvox fucus. Algae are used to men in varieties of the way. At least a half of the total carbon dioxide fixation on the earth is carried out by the algae. Photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of parameter important paramount importance as primary producer of energy rich compound which form the basis of food cycle of all aquatic animals. Many species of Porphyra, Laminaria and Sargassum are among the 70 species of marine algae used as a food. Certain marine brown and red algae produce large amount of hydrochloride, water holding substance. Example: algine brown algae and carrageen red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Glaciaria, are used to grow microbes and in preparation of ice cream jelly. Chlorella, a unicellular algae rich in protein, is used as a food supplement even by the space travelers. The algae are divided into three main classes: chlorophyce, phaeophyce, rhodophyce. 3.1.1 Chlorophyce. The members of chlorophyce are commonly called green algae. The plant body may be unicellular, colonial, filamentous. They are usually grass green due to the dominance of pigment chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage body called pyrenoid located in the chloroplast. The pyrenoid contain protein-based structure. Some algae may store food in form of oil droplet. Green algae usually have rigid cell wall made up of inner layer of cellulose and an outer layer pectose. Vegetative reproduction usually takes place by fragmentation. Asexual reproduction is by flagellated zoospore produced in zoosporangium. The sexual reproduction show considerable variation in the type and formation of sexual cell, and it may be isogamous, anisogamous or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Ulothrix, Spirogyra, and Cara, figure 3.1a. Phaeophyce. The member of the Phaeophyce or a brown algae are found in primarily in marine habitat. They show great variation in size and form. They range from simple branch filamentous form Eucalyptus to profusely branch form as represented by kelp, which may reach a height of 100 meter. They possess chlorophyll A and C, carotenoid, xanthophyll. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment. Fucoxanthin present in is stored as complex carbohydrate which may be in the form of lamarine or mannitol. The vegetative cell have a cellulosic wall usually covered on the outside by a gelatinous coating of algin. The protoplast contain is to plastid, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk, the stipe, and the leaf-like photosynthetic organ. The frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in the most brown algae is by biflagellated zoospore that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gamete may take place in water or within the Oogomus species. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common forms are Ectocarpus, Dictyota, Laminarin, Sargassum, and Fucus. Figure 3.1b.
The member of Rhodophyceae are commonly called red algae because of the predominant of red pigment are phycoerythrin in their body. Majority of the red algae are marine with greater concentration found in the warmer area. They occur in both well-lighted region close to the surface of the water and also at a great depth in ocean where relatively light penetrate. The red thalli of most of the red algae are multicellular. Some of them have a complex body organization. The food is stored as a Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spore and sexually by non-motile gamete. Sexual reproduction is oogamous and accompanied by a complex post-fertilization development. The common member are Polysiphonia, Polyphyra, figure 3.1c, Gracilaria and Geladium. Table 3.1 Diverse division of algae and their main characteristic. Class Chlorophyceae, common name green algae, major pigment chlorophyll A and B, stored food starch, cell wall cellulose, flagellar number and position of insertion 2 8 equal apical, habitat freshwater, brackish water, salt water. Phaeophyceae, common name brown algae, major pigment chlorophyll A, C, fucoxanthin, store food mannitol, laminarin, cell wall, cellulose and algin, flagellar number and position of insertion 2 unequal laterally, habitat freshwater rare, brackish water, salt water. Rhodophyceae, red algae, major pigment chlorophyll A D phycoerythrin, store food Floridian starch, cell wall, cellulose, pectin and polysulfate esters, flagellar number and position of insertion absent, habitat freshwater, some, brackish water, salt water, most. 3.2 Bryophyte. Bryophyte include the various mosses and liverwort that are found commonly growing in moist shaded area in hills. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid and shaded locality. They play an important role in plant succession on bare rock and soil. The plant body of bryophyte is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoid. Back to root, stem, or leaves. They may possess root-like, leaf-like, stem-like structure. The main plant body of bryophyte is haploid. It produces gamete, hence it called gametophyte. The sex organs in bryophyte are multicellular. The male sex organ is called antheridium. They produce bifurcated anthrozoid. The female sex organ called archegonium is flash-shaped and produce a single egg. The anthrozoid are released into water where they come in contact with archegonium and anthrozoid fuse with the egg to produce the zygote. The zygote do not undergo reductional division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derive nutrition from it. Some cells of the sporophyte undergo reductional division, meiosis, to produce haploid spore. The spore germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous plant, herbaceous mammal, bird and other animal species of sphagnum. A moss provide pit that have long been used as a fuel and as packing material for the transshipment of living material because of their capacity to hold water. Mosses, along with lichen, are the first organism to colonize the rock and hence are of great economic importance. They decompose rock, making the substratum suitable for the growth of higher plant. Since the moss form dense mat on the soil, they reproduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverwort and mosses. Liverwort. The liverwort grow usually in moist, shady habitat such as bank of stream, marshy ground, damp soil, bark of tree, and deep in wood. The plant body of liverwort is thalloid. Example Markensia. The thallus is dorsiventrally and closely appressed to the substratum. The leaf member have tiny leaf-like appendages into raw on the stem-like structure. Asexual reproduction in liverwort take place by fragmentation of the thalli or by the formation of the leafy member have tiny leaf-like appendages in two rows on the stem-like structure. Asexual reproduction in liverwort take place by fragmentation of thalli or by the formation of specialized structure called gemma. Single gemma. Gemmae are green multicellular asexual bud which develop in small receptacles called gemma cup located on the thalli. The gemma become detached from the parent body and germinate to form new individual during sexual reproduction. Male and female sex organ are produced either on the same or on a different thalli. The sporophyte is differentiated into a food seta and capsule. After meiosis, spores are produced within the capsule. The spore germinate to form free living gametophyte. Mosses. The predominant stage of life cycle of mosses is the gametophyte, which consists of two stages. The first stage is protonema stage, which develop directly from the spores. It is creeping, green, branch and frequently filamentous stage. The second stage is leafy stage, which develop from the secondary protonema as a lateral bud. They consist of upright slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branch rhizoid. This stage bear the sex organ. Vegetative reproduction is mosses is by the fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organ antheridia and archegonia are produced at the apex of leaf shoot. After fertilization, the zygote develop into sporophyte, consist of a food seta and capsule. The sporophyte in mosses is more elaborate than that in liverwort. The capsule contains spore. Spore are formed after meiosis. The moss have an elaborate mechanisms of spore dispersal. Common example of mosses are Funaria, Polytrichum, and Sphagnum. Pteridophyte. The pteridophyte include hostel and fern. Pteridophyte are used for medicinal purpose and as a soil binder. They are also frequently grow as ornamental. Evolutionary, they are first terrestrial plant to possess vascular tissue, xylem phloem. You shall study more about the tissue in chapter 6. The pteridophyte are found in cool, damp, shady place through some may flourish well in sandy soil condition. You may recall that in bryophyte the dominant phase in the life cycle is gametophytic plant body. However, in pteridophyte, the main plant body is a sporophyte which is differentiated into true root, stem and leaf. Figure 3.3. This organ possesses well differentiated vascular tissue. The leaves in pteridophyte are small microphyllous or large macrophylls, as in fern. The sporophyte bear sporangia that are sustained by a leaf-like appendages called sporophyll. In some cases, sporophyll may form distinct compact structure called strobili or cone. Selaginella equipsitum, the sporangia produce spore by meiosis in spore mother cell. The spore germinate to give rise to inconspicuous small but multicellular free-living, mostly photosynthetic thalloid gametophyte called prothallus. The gametophyte require cool, damp, shady place to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophyte is limited and restricted to a narrow geographical region. The gametophyte bear male and female sex organ called antheridia and archegonia respectively. Water is required for transfer of antherozoid, the male gamete released from antheridia, to the mother of the archegonia. Fusion of male gamete with the egg present in the archegonia result in formation of zygote. The zygote thereafter produce a multicellular well differentiated sporophyte which is dominant phase of pteridophyte. In majority of the pteridophyte, all the spore are of similar kind. Such plant are called homosporous. Genera like Selaginella salvinia, which produce two kind of spore, macro that is large and micro that is small spore, are known as heterospore. The megaspore and microspore germinate and give rise to a female and male gametophyte respectively. The female gametophyte in this plant are retained on parent sporophyte for variable period. The development of zygote into young embryo take place within the female gametophyte. This event is a precursor to the seed habitat, considered an important step in evolution. The pteridophyte are further classified into four classes: Psilopsida, Psilotum, Lycopsida, Selaginella lycopodium, Sphenopsida equipsitum, and Pteropsida, Dryopteris teres adiantum. Gymnosperm. The gymnosperm, gymnonaked sperm seed, are the plant in which the ovule are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seed that develop post fertilization are not covered, are naked. Gymnosperm include medium-sized tree or tall tree and shrubs. Figure 3.4. One of the gymnosperm, the giant redwood tree sequoia, is one of the tallest tree species. The root are generally taproot. Root in some genera have fungal association in the form of mycorrhiza pinus, while in some other cycas, small specialized root called chloroid root are associated with nitrogen fixing cyanobacteria. The stem are unbranched cycas or branch pinus or cadrus. The leaf may be simple or compound. In cycas, the pinnately leaf persists for a few years. The leaves in gymnosperm are well adapted to withstand extreme of temperature, humidity, and wind. In conifer, the needle-like leaf reduces the surface area. The thick cuticle and sunken stomata also help to reduce water losses. The gymnosperm are heterosporous. They produce haploid microspore and megaspore. The two kinds of spore are produced within the sporangia that are borne on sporophyll, which are arranged spirally along an axis to form lax or compact strobili or cone. The strobili bearing microsporophyll and microsporophyll are called microsporangiate or male strobili. The microspore develop into a male gametophyte generation which is highly reduced and confined to only a limited number of the cell. This reduced gametophyte is called pollen grain. The development of the pollen grain take place within the microsporangia. The cone bear megasporophyll with ovule and megasporangia are called macrosporangiate or female strobili. The male or the female cone or the strobili may be born on the same tree, Pinus. However, in Cycas, male cone and the megasporophyll are born on different tree. The megaspore mother cell is differentiated from one of the cell of the nucellus. The nucellus is protected by envelope and the composite structure is called an ovule. The ovule are borne on megasporophyll which may be clustered to form the female cone. The megaspore mother cell divide mitotically to form 4 megaspore. One megaspore enclosed within the megasporangium develop into multicellular female gametophyte that bear 2 or more archegonia or female sex organ. The multicellular female gametophyte is also retained within the megasporangium. Unlike bryophyte and pteridophyte, in gymnosperm, the male and the female gametophyte do not have an independent free living existence. They remain within the sporangia written on the sporophyte. The pollen grain is released from the microsporangium. They are carried in air current and come in contact with the open ovule borne on megasporophylls. The pollen tube carry the male gamete, grow toward archegonia in the ovule and discharge their content near the mouth of the archegonia. Following fertilization, zygote development in an embryo and the ovule in the seed, these seeds are not covered. Angiosperm. Unlike gymnosperm where the ovule are naked in the angiosperm or the flowering plant, in angiosperm, the seeds are enclosed in fruit. The angiosperm are an exceptionally large group of plants occurring in wide range of the habitat. They range in size from the small Wolfia to the tall eucalyptus over 100 meter. They provide us with the food, fodder, fuel, medicine, and several other commercial important products. They are divided into two classes, dicotyledon and monocotyledon. Summary: Plant kingdom include algae, bryophyte, pteridophyte, gymnosperm, angiosperm. Algae are chlorophyll-bearing, simple thalloid, autotrophic, and largely aquatic organism. Depend on the type of the pigment possessed and the type of store food. Algae are classified into three classes, namely chlorophyce, phaeophyce, rhodophyce. Algae usually reproduce vegetatively by fragmentation, asexually by the formation of different type of spore, and sexually by the formation of gamete which may show isogamy, anisogamy, or oogamy. Bryophytes are the plant which can live in soil but are depend on water for sexual reproduction. Their plant body is more differentiated than that of the algae. It is thus it is thallus-like and prostate or erect and attached to the substratum by the rhizoid. They possess root-like, leaf-like, and stem-like structure. The bryophytes are divided into liverwort and mosses. The plant body of the liverwort is thalloid and dorsiventrally, where mosses have upright, slender, axis-bearing, spirally arranged leaf. The main plant body of the bryophyte is gametophyte producing and is called as gametophyte. It bears the male sex organ called antheridia and the female sex organ called archegonia. The male and the female gamete produce fuse to form zygote which produce a multicellular body called a sporophyte. It produces haploid spore. The spore germinate to form the gametophyte. In pteridophyte, the main plant is a sporophyte which is differentiated into true root, stem, and leaf. These organs possess well differentiated vascular tissue. The sporophyte bears sporangia which produce spore. The spore germinate to form gametophyte which require cool, damp, antheridia and cool damp place to grow. The gametophyte bear male and the female sex organ called antheridia and archegonia respectively. Water is required for transfer of male gamete to the archegonia where zygote is formed after fertilization. The zygote produces sporophyte. The gymnosperm are the plant in which ovule are not enclosed by any ovary wall. After fertilization, the seed remain exposed and therefore these plants are called naked seeded plant. The gymnosperm produced microspore and megaspore which are produced in microsporangia and megasporangia borne on sporophyll. The sporophyll, microsporophyll, and megasporophyll are arranged spirally on axis and pollen tube release the male gamete into the ovule where it fuses with the egg cell in archegonia. Following fertilization, zygote develop into embryo and ovule into the seed. The angiosperm are divided into two classes, dicotyledon and monocotyledon. Chapter end. Thank you.